I am Dr. Khokon Kumar Dutta. My Christian name is Dr. David Moon. I'm working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is MicroRNA-34a, the master regulator of cellular and organ regeneration, the beginning point of the great story. MicroRNA-34a family has three members: MicroRNA-34a, MicroRNA-34b, and MicroRNA-34c. In 2003, American Nobel Prize winner Professor Dr. Philip A. Sharp and his group for the first time reported that MicroRNA-34a is expressed in the mouse embryonic stem cells. Stem cells seems to be of different types depending on their potentials and characteristics. For example, totipotent stem cells are capable to give rise to all types of cells of human body and other life forms. But lineage-specific stem cells can make only a particular types of cells. There are different other types of stem cells. The discovery of the presence of MicroRNA-34a in the embryonic stem cells may suggest that its function may be related with the behavior of stem cells. That means with proliferation or maintenance of the stem cell pool. Many genes functions are needed for the stem cells. Some of them work as housekeeping genes that also contribute similarly in the differentiated cells of any lineages. And some genes functions are only needed for the stem cell-like characteristics, whose functions are not found in the mature cells. In our work with MicroRNA-34a in the School of Medicine, Kyoto University in Japan, we found important role of MicroRNA-34a in cancers. The main danger of cancers come from the spatial characteristics of uncontrolled proliferation of cancer cells. Abnormal growth characteristic is not unique for cancer cells only. There are different other diseases where growth abnormalities are seen for the cells, which are very close to the normal ones. But the cancer's growth makes sharp differences with those types. In our experiments, we tried to use the functions of MicroRNA-34a to relate actively proliferating cancer cells and the stem cells. We introduced the possibility of the presence of important roles of MicroRNA-34a in the intrinsic dedifferentiation or dedifferentiation process, which works as the key factors, at least in the carcinogenic process of well-differentiated cells or cells with maturation of any degrees. Proliferation and regeneration processes have some differences in between them. In proliferation, the cells may increase in number, or in size, or in both. But regenerative proliferation specifies different conditions where resting cells start proliferation again to replenish some losses usually. Regeneration is a specific type of proliferation. Sometimes we face difficulties to understand another process. Rejuvenation frequently comes with the previously mentioned two processes. Rejuvenation is a work in the opposite direction of changes in the cells or in anything of this universe. Changes makes elements of the materialistic world a subject of a dimension called time. Rejuvenation can be defined in a simplistic way as the walk backward the arrow of time. Anyway, in our experiments, we use certain chemicals, ferric nitrilotriacetate, FeNTA, to induce damage in the rat kidney. After a certain time of inducing damages, we noticed regeneration in the kidney tissues. We found high levels of MicroRNA-34a expression in the regenerating cells in the damaged tissue in the rat kidney. This was the first experiment internationally that connected MicroRNA-34a with the regeneration process directly. The observations was published in the journal Cancer Science in December 2007. The report can be found in the volume 98 and pages from 1845 to 1852. Till 2020, scientists around the globe succeeded to explore much of the important involvement of MicroRNA-34a with different biological events in normal physiology and in disease conditions. MicroRNA-34a is now known as the master regulator of the cellular and organ regeneration process. Some scientists are even demanding this very small RNA molecule to hold the capabilities or potentials enough to influence the regeneration process, like Prometheus. Some of the advancements relating MicroRNA-34a and organ regeneration can be mentioned here to validate the importance. Xiaoping Wang et al. Published an article in the Journal of Oncotarget in February 2017 with the title MicroRNA-34a regulates liver regeneration and the development of liver cancer in rats by targeting NOS signaling pathway. Yanpei Yang et al. Published an article in the Journal of Circulation Research in August 2015 with the title MicroRNA-34a plays a key role in cardiac repair and regeneration following myocardial infarction. Huan Liu et al. Published an article in the Journal of Stem Cell Research and Therapy in June 2019 with the title MicroRNA-34a promotes bone regeneration in irradiated bone defects by enhancing osteoblastic differentiation of mesenchymal stromal cells in rats. Yang Jin Cho et al. Published an article in the Journal of Nature Cell Biology in October 2011 with the title MIG-34a microRNAs provide a barrier for somatic cell reprogramming. Michael A. Bellio et al. Published an editorial in the Journal of Annals of Translational Medicine in June 2016 with the title Is the regulation of SHRT1 by MicroRNA-34a is the key to mesenchymal stem cell survival. It is to mention here that mesenchymal stem cells play important roles in the regeneration process of different tissues in the human body. It can be expected that detailed exploration of the functions of MicroRNA-34a may even help someday to materialize the dream of scientists worldwide to be able to regrow the lost limbs of human body that makes a huge painful burden to all the countries today. Finally, I would like to wish happy, prosperous, and dignified life for you all. Thank you, everybody.